Good evening and welcome. My name is Michael Lonsborough and I'm a British scientist working in the Czech Republic and also the host for this evening's Global Science Café Czech Centres. Just a few words at the beginning about Czech Centres. Czech Centres is a government-funded organisation and part of the Ministry of Foreign Affairs of the Czech Republic. As such, it's a core instrument in foreign diplomacy for the Czech Republic, and also it does this by promoting Czech science, Czech arts, and Czech innovation and creative industries through 25 branches across 3 continents of our planet. Part of this remit is, of course, the Global Science Café, a series of lectures and debates with top Czech scientists, innovators, and professionals. And this evening is the third in the series of these Global Science Café Czech Centres, and it's my privilege to welcome you to our guest for this evening. It's Dr. Michal Šimíček. Welcome.
Good evening.
And Michal Šimíček is an award-winning immunologist and molecular biologist who, after working at prestigious universities in Prague, in Belgium, London and Cambridge, has decided to return to his hometown of Ostrava, where he's established a new group looking into the innovative science of cell therapy for oncological diseases. Michal, once again, welcome, and maybe we can start at the beginning. Who is Dr. Michal Šimíček?
So I'm from Czech Republic, from Moravia, where I was actually born. I sort of grew up in a tiny village under the mountains, surrounded by nature, in a kind of small farm. Then, you know, through the time, I was getting more and more fascinated by the processes like happening around me, and this sort of brings me to my, after like of course long journey, to my current place. You know, so being a researcher scientist, or even like a manager of a scientific team, trying to develop a new therapeutical product, understand how the biological processes work in the pathological settings, and ultimately bring some new hope, some new treatment for patients with cancer.
Okay, that's quite a story, and that's. I'd like to sort of divide that story up into little chunks. Let's begin at the start. So you mentioned you grew up on a farm in the north parts of Moravia,
Yeah, that's true.
and you're sort of a village life, lots of the outdoor living. But tell me a bit about the spark that got you into science. How did you go from somebody living on as a young boy on a farm to really getting into science and understanding all about those processes that you?
I would say it was pretty much natural for me. You know, there was no particular person that would dedicate me to do science. I was sort of like, even as a small boy, exploring all the moving stuff, or even like stones around me. I was quite fascinated by the processes, how the nature works in system like ecology, and then later on, when I understood more about chemistry, how the building blocks of the matter, so atoms and molecules, work together to sort of make us a human being. Yeah. So for me, it was, as I say, sort of bits and pieces throughout my life, especially my childhood, that sort of dragged me into the field of science, that I later on became more or less like in love, you know.
So almost an inherent interest
Indeed.
in natural processes, in the science of things, and that turned to sort of a love for science. From when you leave after schooling in Ostrava, you then go to Prague University.
That's true. Actually, I sort of studied not directly in Ostrava, but in my village, in a nearby town, because I'm not directly from Ostrava. I'm, as I said, from like mountains nearby. Then indeed I moved to Prague for my uni. It was one of the best times of my life. You know, student life, and Prague at that time was really cool. Maybe now it's even better. Can't compare. Then I had a chance to spend some time in London. So this sort of bring me opportunity to learn and improve my English, because originally I studied more German, and English, you know, was more or less thing that I had to get like in a run, you know. So I wanted to do the science, and the requirement was you must know English. There was no other way, right? So basically, I went to London. I could speak like, just express myself really, really in a bad way, but I had no choice, right? I was like a non-swimmer in the sea, and after a couple of months, I basically got to know the basic stuff, and to get even better in the language, and also to see science from different points of view. I decided to go for a Ph.D. for doctoral studies to somewhere else than somewhere to abroad. So I applied for an PhD, international PhD position in Belgium. Was selected and got a position there. And it was a very influential time there, because my boss is actually a holder of several important European grants. So I could see how these applications are prepared, how to get grants like ERC and similar.
So can I just jump in quickly, because I'm aware that this evening there might be several, or many, I hope, younger members of the audience listening in across Facebook, who might be interested in your journey, that educational process. They themselves might be in their first degree, a science degree perhaps, or other, and so I'm sure they'd be keen to hear something about the challenges that were presented in your first degree. So your first degree was in immunology, specifically.
Right, that's it. Yeah.
Right, and so tell me a bit, was it difficult then you mentioned that you finished the degree in London, because you realized you needed to get a good grasp of English if you wanted to become an independent scientist.
Exactly.
So tell us a bit about the problems you faced in that transition.
So as I said, I guess for the current generation, English is not a big deal. They basically get it. They learn it from the very beginning of a school. So the biggest challenge for me, I graduated from immunology, but then for the doctoral studies, I moved to a slightly different field. So which is cancer biology. So I had to obviously study many new articles. Like I had to get into different techniques and be open to like the ideas and comments from different fields than I was sort of raised in as a pre-graduate student.
And when you're making these decisions about fields and selecting which are the right modules to study, what's guiding you here now? If I remember my university days, is it books you're reading? Is it individual lecturers who you have a better liking for, or a greater interest in their specific work, or are you following sort of trends coming from the scientific literature? What's the guiding principle there?
So for everybody it can be different. For myself, I was since end of the first year at the uni, I was working in the Institute of Molecular Genetics here in Prague as a part-time scientist or junior scientist. So I had a chance to be in the real scientific lab already from the very beginning. So I could basically confront the theory that I gained in the lectures with the practice, with the real scientific life in the lab. So this was very inspirative for myself, that I could indeed see how the theory can be applied. I could do experiments myself, which was very fascinating. And nowadays there is a huge bench of information online that actually wasn't when I was the pre-grad, So indeed I had to stay in the library, study a lot, and but what sort of moved me forward and give me this this sort of new passion, or the bigger passion for science, were the discussions with actual scientists. That in the lab, or even afterwards in the pub, or
that's right. These intellectual sort of deep debates about what an interest, a point of interest. I completely agree. So in London, I can imagine now. So you're a young man, whether it be in Prague, in London, later on in Belgium. Then you're having these wonderful scientific debates with mentors, with fellow students from the lab, in the pub. All this is leading to a sort of a gathering momentum in your intellectual development, and a gathering. Are you at this point? Are you becoming more and more sure that a career in academia and a career in research is the right thing for you?
Right. That was exactly as you described. Basically, even nowadays, that's what I like the most. This sort of brainstorming debates with students or professors when we just put ideas on the table. We just have like sometimes really crazy hypotheses, but some of them turn out to be true, or you're just being basically fascinated by all these things that you can come up with and then test in the lab, and later on show that it's indeed as you saw. And
so you're having, is it fair to say, the time of your life in your Ph.D. in Belgium? You successfully defend your thesis, and at which point then you're looking for somewhere to do some postdoctoral studies, with the hope presumably then to continue into academia. And tell us what happens then.
Indeed. So I was basically lucky to be selected as one of the only student giving a talk in Cold Spring Harbor, which is a place in US near New York, where is a quite famous biological campus. So Watson Crick and these guys were working there. So basically, it's a place where they did describe the double-stranded helix, double-stranded structure of a DNA. So I gave there a lecture, and it was a place where I met my future boss. So it was actually a guy from Cambridge working in the field or with the molecules that I was particularly interested in during my PhD, and he offered me the position in the lab, which was slightly, was not literally what I did in my past. It was not immunological lab and not a lab focused on cancer, but they were more into biochemistry and structural biology, which is something that I didn't touch a lot during my previous career. So it was something that I should consider whether to go there or not.
So it's also a step of faith from this scientist in Cambridge, looking from somebody who's not necessarily directly from his field, but understanding that you've got something to contribute, coming from your background in immunology, and then afterwards doing structural molecular biology, presumably.
Indeed. They were actually looking for some cell biologist, so somebody that could bring their molecular findings into the actual living creatures.
Right, somebody who could understand the structural implications of certain molecules and how they can do their work inside the cell environment.
Exactly. And I had other interest. I wanted to know how basically the actual mechanistical features of the molecules imply into my in the features of the cell behaviors that I was studying during my career. Okay. So I wanted to basically connect these two fields, understand how the actual mechanism works on the atomic structural level.
Let me just stop you then. Let's for the benefit of our audience, tell us a bit about how does one, it's completely understandable, this idea, this concept of understanding the functions and the structures of individual molecules in a cellular environment. Tell us, we're talking, these are of course, the molecules are nanometers big, the cells are micrometers big.
Right.
Tell us a bit about how does one do that?
Well, there's many approaches, but if you are literally non-experienced person coming to the lab, you sometimes might even laugh, because it looks like you are just moving tiny drop from one place to another. Or
the classic pipette.
Right, exactly. That's or moving one paper from one basket to another basket, and just this kind of crazy things that people would never see something deeper behind it. But when I say that I work with cells, usually people think that you are just sitting the entire day behind a microscope, looking how the cells are moving there, what they do, and these kind of things. But it's just there's a little bit of the truth, right? Because most of the work is with these tiny drops or measuring some spectral features, or things that take a relatively long time and are pretty boring on the way how they are done. But what's exciting is the actual result at the end. That you reach, and that you can basically interpret, connect with other results to see the big picture, how the molecules are working in the cell, and how they how the orchestra of their function basically then relate or represent the particular feature, like cell division, cell movement, or transformation into cancer and similar.
So these skills that you've developed during your PhD are now being applied in Cambridge, specifically looking at cancerous cells.
Indeed. We're looking at mainly at the cancer cells, how the cancer develop, how when the cell acquires particular mutations, what happens. But we had also side projects looking into neurodegenerative diseases like Alzheimer's disease and similar. But at the level that we were working, basically. We basically study particular molecules that could be associated with either or with both types of diseases, so cancer, neurodegenerative, and so on. So it was more, let's say, basic, fundamental research.
Okay, so let's take another step back and think about, because I'm concerned that we're going to be using terms, increasingly more terms, which might be slightly incomprehensible to many. So from now that we're into your postdoctoral career, where you're looking at the effect of various molecules on cancerous cells, concentrating then into certain systems of the body, certain tissues, and indeed the hematopoietic system. Let's maybe begin this discussion with a word that we're all familiar with, which has already been heard, and that's cancer and cancerous cells. So tell us a bit about your understanding of what cancer really is, and if we can start there, then go on forward to the more specifics.
Sure. So I would like to highlight or point that cancer is just one word, but it's tens or hundreds of different diseases. Right. So so one common thing that the cancer cell have or feature is basically uncontrolled growth. The cells can just be sort of immortal. They can divide and proliferate. The scientific word is proliferation. They basically grow, and they even escape from the tissue they originate. And they form metastases all over the body, and the source of the cancer is usually some kind of mutation in the DNA. So it's basically genetic disease. Cancer is disease of DNA, to be kind of precise.
And in terms of ballpark figures, such diseases, as you pointed out, there are hundreds of cancers, of diseases which can be termed cancer. But if we put those all into one group, that affects what sort of portion of the human species at one point in their life? Yeah,
it's a good question, because we have nowadays kind of feeling that the cancer is being more and more frequent, which is actually true. But to be honest, scientists are quite surprised that we don't develop cancer much earlier in the life in general. And the reason is that, as I said, it's a disease of DNA. It's caused by mutations, and the mutation can occur either or can be caused by some external factor, as a pollution, smoking, you have like a bad life habit, but also it could be some inherent process in the cell, right? So basically, we all are breathing oxygen, which is important for our metabolism to create energy in our cells.
Yes. And
during the process of creating energy in the intracellular organelle called mitochondria, maybe somebody knows, is a side product of this metabolic pathways are reactive oxygen species. So there are atoms or molecules which that have ability to damage DNA and create mutations.
That
Right?
reminds me a bit of at school, my chemistry master used to always describe oxygen as this beautiful, knock-dead, gorgeous lady who's willing to react with almost anything, and therefore, where it gives us life and we use it so importantly in our metabolisms, but then at the same time it has the possibility to react with parts of our body and organelles and DNA that we don't particularly want to happen. Right. Okay. So these cancer, and of course, cancers can, as you said, in any age. Isn't it the case that almost every day there is the potential for cancerous cells to develop, and yet healthy, especially younger, healthy individuals are quite capable with their own immunology to shut down that process and get rid of those cancerous cells.
Indeed. I was basically going to this point that the metabolism basically occurs in every cell of our body, so damage can be anywhere, and there is indeed damage. Every day, many cells become sort of precancerous. So the body has to have some kind of hammer, some way how to get rid of these cancer cells. And one of the main mechanisms is based on the immune system, our immune system, our immune cells that are able to really quickly detect and destroy the cells that have even just potential to become cancerous.
Right. So these are, from my ignorant perspective as a chemist and having very little knowledge of medicine and our anatomy per se, these are sort of white blood cells, these T cells, which are in circulation in our blood system, percolating all our tissues. They're all the time looking out for such potential malignancies or potential cancers.
Exactly. As it's precisely as you described, that we have these white blood cells, but many different types. Not just the T cells, T lymphocytes, but also some kind of natural killer cells or NK cells that are like dedicated to eliminate cells which are infected by viruses or the cells that have this malignant capacity.
And how do they do that?
There is several mechanisms, but for NK cells, it's basically a recognition of some change. Something basically there. So every cell has some molecules on their surface that indicate their state. That basically they belong to a particular body, that are mine or your.
Yes.
That we cannot basically share the cells because the immune system would recognize them as something foreigner and basically reject them. And they basically present this kind of "don't eat me" signal. That they basically say, "Hey, NK cell, I'm healthy, I'm fine. You basically can go and check my neighbor," right? But when the cell becomes cancerous or infected by virus, these molecules can be changed or something can occur. So when there is some discrepancy, the NK cell doesn't care and just kill the cell immediately.
But in terms of molecular, how does it kill it?
It's a pretty simple mechanism. It literally expels or expels some kind of enzymes that make a holes.
So it just blows a hole in the cell.
Indeed. It just blows the holes.
And the content of it just comes.
Indeed,
extrudes out into the. Yeah. So that's fascinating. Okay, so you're in Cambridge. You know a lot about. You're gaining more and more knowledge about cancer. You've got this molecular biological background, and you're going forward. Now you begin then to sort of look in particular at the hematopoietic system. So tell me, please, what is the hematopoietic system? What it does for us, and how does it become how do malignancies occur there?
So as we already touched, the hematopoietic systems are both white and red blood cells in brief, and then you can distinguish many different types of the blood cell types. So if I would be focusing on the immune system, so in addition to these T cells and NK cells which we mentioned, there are also B cells and different other granulocytes, neutrophils, and so on, with different functions in protecting our body against the pathogens or the cancer cells. And as any other tissue in our body, also these immune cells each of their kinds can during our life become malignant, become cancerous. So with all the consequences that are well known.
Right, and at this period of your intellectual development at Cambridge, what was the state of the art? How did we, in terms of doctors, at medicinal facilities, at hospitals, working with patients suffering from these types of cancers, what was the state of the art? How were they treated at the time?
So it's not so far in the past, yeah. So it's basically still more or less the same nowadays. Even it's slightly changing, slowly changing. So, the current treatment of cancer is based on applying different kinds of chemotherapy, radiotherapy, combining with more modern therapeutic approaches based on antibodies or the actual modified cells, which we will probably talk later on.
Because, in terms of just using sheer logic, if we understood for a reasonable amount of time that there's our own immune system is able to deal with many cancers at their beginnings, and yet then we go to sort of these chemotherapies and radiological therapies, which seem to be quite a large, big, heavy hammer to be hitting a patient with in a metaphorical sense. Why didn't we immediately, first of all, go to, we've got the immune system, how can we learn from that and maybe augment the capabilities of that? Because I think that's eventually where then you go into that system, right? That's the path that you go to follow.
Exactly. This is basically what we are trying to do, after many decades studying the human immune system, how it works, how the cell looks like, what molecules are present on the membrane of the cells, and so on, we can finally use this knowledge to develop therapy which is based on this already like a natural system that our body has, right? So basically creating the anti-cancer immune system, like let's say artificial, which we can then later on apply to patients.
Now, the source of this knowledge, I'm presuming this must be a huge field in science, in scientific research. There must be literally hundreds of scientific groups at top universities looking generally into our immune system, immunology.
I think even more, yeah.
Yeah, so it must literally mean that every day you're getting good quality publications coming into the literature, which you have to keep on top of
course,
presumably. Yeah. So that's I should imagine you spend a long part of every day keeping tabs with this development, this body of knowledge about our immune system. Now, tell me about, with all this, continually updated knowledge of our immune system, what specifically are you doing to augment that? Let's go into a few details now of how you are contributing to the development of cell therapy for these oncological diseases, as opposed to radiology and chemotherapy that was...
Okay, so the whole concept of a cell therapy is, as we already discussed, based on using the immune cells either directly from the patient or in the future from any or even unrelated donor. So we literally take these immune cells from the patient or from the donor. In the lab, we modify them. We basically insert new genetic information into the cell in order to provide them with a gene that encodes a protein molecule that acts as a sort of receptor that can recognize specific surface marker, specific surface feature of a cancer cell. So it can basically drug the immune cell near the tumor where it specifically destroys. So and what's the difference compared to chemo and radiotherapy? Well, obviously it's based on cells, but the main advantage is the dramatically higher efficacy and also the specificity.
Right.
That the immune system kills, let's say, 99% the cancer cells. the chemo and radio, it kills basically like cells that have some kind of similar features. It grows a lot, or it has a
High metabolic rate.
high metabolic rate, indeed, and this kind of things, right? So it's not only the cancer cells, but it can be cells of hair, you know.
I see. So with your cell
epithelial
therapies, you're
so-
able to get a sort of
Right.
specificity of 99%. So you're able to really
it's very high.
hone
It's
in
very
and,
high.
and concentrate the effects of this treatment specifically just on the cancerous cells.
Well, it
Well,
depends on the type of the tumor. You know,
okay.
tumors are very variable, but we can reach very high efficiency and specificity.
So am I understanding correctly that you've got this, the actual process, what the immune system are, the immune system of a healthy person, what that can do, as we said, you sort of literally bores a hole into the cancerous cell and kills it thereby. What you're doing is you're augmenting this system by genetically modifying it to make it even more effective at getting to the right place in your body.
That's it.
So to detecting cancers, malignancies earlier, presumably, and getting there more efficiently.
Or even like after, let's say, the surgeon resect the tumor, there may be some remnant cells, right? That the chemo or radiotherapy cannot destroy effectively.
Yes.
But by applying these immune cells, you just eliminate them 100%, and that's it.
Fantastic. So, okay, so let's talk a bit about these. In my mind, I've got these pictures now of these, immune cells which have gone through your capable hands in your laboratory, and you've done this genetic modification, and they've come out sort of bigger and stronger and better and shinier and the rest of it. So tell me a bit, how do you do this genetic modification? How do you take these immune cells and give them these almost superhero powers, as it were?
So, like practically, in the past, it was usually based on like infection with viruses. So we create some like a virus that lost the ability to replicate. So it's just a platform to transfer the genetic information into the cell. But for safety concern, nowadays we are moving into more like, let's say, physical techniques. So we use a technology called electroporation. So we literally give an electric shock, electric pulse to the culture that contains the cell. So that what happens in the cell, which you can imagine like a tiny ball, for like nanoseconds there are tiny holes created. So the DNA can like penetrate into the cell. Then the holes are sealed, and the cell happily continues.
That's
its
incredible.
life.
So you're sort of firing almost little electrical bullets into the
indeed,
cell to make
indeed.
little holes through which then fragments of DNA that you want goes in, and the cell is then able to receive those fragments and incorporate them into its own genome.
Right. It can either stay as it is in the cytoplasm, not in the nucleus, or if we provide certain sequences, certain pieces of DNA, then it can indeed go into the nucleus, incorporate into the genome, and stays there for.
And what are the relative advantages and disadvantages of using that viral plasmid technique of using, hitchhiking a virus, getting your DNA fragment into the cell, as opposed to using this electroporation method? Right.
So with the viruses, there is often a risk that it could be incorporated into the places of our genome that we don't want, right? So it can basically activate some genes that can turn the cell cancerous, right? So at the end, we want to provide a cell that will kill the cancer cells, but the immune cells themselves could become cancerous if we activate certain genes. And the virus is integrated usually non-specifically, just jumps into the genome, and that's it. But with the electroporation, we can combine it with other technologies such as CRISPR, a very famous word nowadays, and then we can direct the piece of DNA into particular place in our genome with very high precision and specificity.
Okay, and again, we're talking. Want to remind our viewers, our listeners, that we're talking about nanometer scales here. We're talking very, billionths of a meter, these are not visible to the eye. How can you follow all this? How can you follow where the DNA is going, which genes are coming in, which are what's working, what's not working? How do you follow these things?
Well, of course, there are many different techniques how to do it, molecular biology or genetic techniques. And well, if I could just point on some, yeah, we can literally extract the DNA from the cell and using kind of molecular scissors, so some special enzymes that recognize only certain sequences in the DNA. We can cut these places, then cut these pieces of DNA, compare their size, or we can read the nucleotides. We can read the letters in the DNA sequence and see whether our DNA was inserted indeed into the place where we wanted. So basically, the sequence follows and contains the genes we wanted to put there, and so on and so forth.
If I sort of slightly simplify it somewhat, is all this going on in a test tube in your lab, as it were?
Well, something yes, something not. Like some of the methods are like so sophisticated that we need to outsource them, or we just basically for like small research lab as we are research center, it's not affordable to have a machine for DNA sequencing.
Yes,
right.
Of course.
It's much cheaper basically to order sequencing from some external company that does it for just a few crowns. That's it. For us, it would be hundreds, right, if we do it ourselves. So.
But I mean, you've got a patient, you've taken some of their immune cells for genetic modification. So give me in layman's terms, what sort of quantity of cells are we talking about? These culture of cells, when you're going through the methods of either plasmid type introduction of DNA fragments or the electropurification technique, what's the success rate? If you've got this batch of cells, how many are successfully becoming augmented with this capacity, then which you could then put back into the patient? Sure.
Actually, it depends. It depends on a certain type of cell. For example, these NK cells, they are extremely difficult to be genetically modified. But nowadays, we after a couple of years of a lot of like failures and small successes, we reach already a stage that we can reach something like 50-60% of cells to be modified, which for this particular cell type is really high. For other cells, we are able to reach almost 100%. Yeah, but for this particular cells, we talk something about a half. And if I should say like specific number, well, it's tens of millions. Yeah, tens of millions the cells that are being modified at the initial point, and then of course there's a period of expansion where we upscale, where we make a huge number of these modified cells. We aliquot them, put them into separate tubes. We can cryopreserve, keep them in liquid nitrogen, and then use them as a sort of off-the-shelf product for the patient when it's needed.
Incredible. So you've got to this stage now in your own personal development, in your own build-up of knowledge and know-how, that you are in a position to choose your next step. And your next step from Cambridge was to return to Ostrava.
Right.
So tell me a bit about that. What was complicated about that, perhaps even as well, the hardships involved, the good sides. In the meantime, you're, if I'm not mistaken, you've also got married, become a father. So you're juggling now many balls, families and professional career. So tell me a bit about that transition.
Well, it was really one of the biggest moves in my life. Definitely a huge surprise for many people that expect me to go like slightly different direction or to different place, to be precise. But I'm in Ostrava already for 3 years, and I'm really happy for this decision. Initially, I took it as a challenge, simply as a challenge, that whether I'm able to do something like this, go from place where basically all the or many Nobel laureates are working in you know, when you can basically be co-author on Nature Science on yearly basis, to place where basically the science is like, let's say, in the very like early
Stages,
ages,
yeah.
right? But I saw this as an opportunity to build something new, something that I can develop like according sort of my vision, and I can contribute to the very beginning, which was super exciting. And so it was from, let's say, the professional point of view. Also, there was and still is a huge support from the regional government, from the university. We also actually have some commercial partners in the campus. This is very important for the later transition of the results to the clinic. And obviously, the second aspect for coming back was like my family, the personal reasons that I inherited a farm in our village that I have to take care of. So it's like a big responsibility, and it's kind of like a second part of my life to science. So
I'm a big fan of the English. But I don't know. Maybe you got to play it when you were in Cambridge. Cricket, and indeed the ex-cricket captain of England, Cook, his name was. He also had a farm and managed to juggle his performances on the international cricket stage with running a farm. But so you're in Ostrava. You said you said it was an opportunity for you to realize your vision. So let's talk a bit about your vision in Ostrava and for your team that you've established there. So yeah, the vision, your goals.
So we definitely, I mean, our ultimate goal, in one sentence, is to help patients. That's basically the final goal. And of course, from the lab bench to the patient bed is a long journey, right? That you have to go through. Many people have to be involved, and we start from the very beginning. We do both the fundamental science, just exploring the general mechanism, how the cells work, how they become malignant, how they become resistant to certain drug, then through how we can improve the immune cell to target cancer, and to develop even better therapeutic products that are available at the moment to the last stage. So basically, which will be the actual clinical studies and bringing the product to the patient.
So you're looking at these cancerous cells, you're looking at the chemistry, the actual structural chemistry of them, understanding how you can genetically modify the immune cells such that they can improve their capacity to identify these cells as early as possible, get to them and become effective as early as possible. Once the cell therapy that takes place, or the modification of these cells, once they're ready, can you sort of put them in a bottle and keep them and use them in different patients, or is it very much specific to individuals?
At the moment, actually, the only approved or the type of therapy that can be used only in the clinical studies at the moment only is the one that's based on sort of autologous transplantation. So basically, you can use only the cells from the particular patient, right? Because of the tissue compatibility. But in the very close future, I hope also thanks to our research, we could provide the patients with some kind of universal cells, universal immune cells that can come from different donor, and basically how we create this these universal cells, we kind of shave them. We get rid of the molecules that make them different. So we make them invisible for the patient immune system. It will not recognize them. It will not destroy them. And then basically we can have this off-the-shelf universal product for different patients.
And such universal cells, they can be bottled, right? And presumably they can be transferred from lab to hospital, hospital to different hospitals. Is there do these things travel well?
This is a good question. Compared to like the classical chemotherapeutic products or pills, that can be generated in one place in the world and then distributed. The cells, even they are cryopreserved, they are very fragile and they cannot be moved for a long time and long distances. So they should be, or in the future, I believe there will be some sort of local centers that will be generating the cells in the place and providing to the neighbors hospitals.
I see. So does that mean that your vision for your group in Ostrava is not limited just to Ostrava Hospital, but then essentially you could be in collaboration, in connection with a wider region?
Definitely.
Supplying solutions, cell therapies to many different hospitals.
Definitely. Or our first goal, of course, is to bring the product to our clinic and the neighbors' ones. But in the longer perspective, we would like to transfer the technology that we will develop to the other places, to sort of outsource, so that not only in Ostrava and around it could be done, but also like a bit more in distant places.
Right. Well, at this point, let's have a look at. I can see we've got some questions coming from people who are following us. Question number one from Rachel: Is there a way to use cell therapy as a way of prevention rather than a therapy of prevention? For example, among patients with tumors coming back after treatment has been finished.
Oh yeah, this is great question. I have this sort of provocative term for this, which I called, let's say, cancer vaccine. Yeah, so which is basically cell therapy that is provided to the patients exactly for this reason. Once the tumor was resected, or it's basically not, or the clinicians are not able to detect the residual cells because of the resolution. Even they are there, but with the current technology, we are not able to detect. They can be hidden somewhere, right? For the imaging or different kinds of detection technologies. So, as a sort of prevention, or to get rid of all these remaining cells, the cell therapy or cell vaccine, anti-cancer vaccine, could be in the future provided to the patient to get rid of these residual cell populations, to not get.
So it's sort of a preventative measure from a reoccurrence of the cancer,
indeed.
Which of course is, I think, that's quite a common thing as well, isn't it?
It's very common. It's unfortunately, still a big problem.
Now, there's several questions that are asking you about natural killer cells, which you briefly mentioned within our immune response, and they're asking about whether there are actual clinical trials using these natural killer cells in the fight against cancer, and especially they're asking with reference to cancers of the breast.
Yes, actually, NK cells are pretty new in the cell therapy field, but even that there are, to my knowledge, at least 2 clinical trials. In the world, and the results, the current results seem to be pretty promising. Hopefully, we want to start our own studies soon. So when we get the products ready, we are at the moment just at the preclinical, or already at the preclinical stage, meaning basically testing the cells in the animal model. Seems to work quite good. So hopefully we can join the team. And the second part of the question, whether it could be used to treat different types of cancer than cancer of the hematological system that we are primarily focused on. The answer is probably yes, but not yet. We are trying to improve the technology or the cells in a way that we can also direct them towards a solid tumor.
And if you can sort of briefly answer why is it that these natural killer cells are highly functional against certain cancers and others not so much?
Honestly, it's not really known. The one of the explanation is that basically hematological cancers, so cancer of the blood system, are not forming like a very solid tumor mass.
Yes.
They are basically like liquid.
Yes.
Right?
Yes.
So the cell can literally meet each other and
easily. Of course, it's a concentration thing.
Because for the immune cell to kill the cancer cell, there isn't like direct contact needed.
Yes.
You know, they have to literally kiss each other, and
Yes.
you know, the bullet should be shot.
So a tumor can only affect on the surface, then it has to go again and again and again, and so it might be slower than the actual tumor growth.
Exactly. But for the solid cancers, I see the more likely application after the surgical resection. So once the tumor is removed, then obviously there are some cells remaining. So we can provide the NK cells to the patients to get rid of the residual cells, right?
So there is hope. There is hope there. On the subject, we remain on the subject of these natural killer cells. There's a question asking whether the natural killer cells, or presumably maybe even the natural killer cells that you can genetically modify, would they be useful in for treatment with patients with COVID-19?
It's a good question, because actually the natural killer cells, the NK cells, they're, as I mentioned in the beginning, their main function is to eliminate cells which were infected by viruses. So in principle, they could be. The only thing with COVID is that it's really quick. It's very quick, and basically once you have the symptoms, once you are diagnosed with COVID, it may be too late to provide the NK cells, because the body is already, or the lungs are already over-infected. Yeah, so you can potentially use it as a prevention, as a vaccine, but this is probably too optimistic, and it will be. I can't imagine the cost of this vaccine.
Well, yeah, I read recently that COVID-19 is meant to trigger some sort of bradykinin storm, a huge release of a certain protein which has an effect in multiple tissues and leaves certain tissues open to attack from other pathogens or other problems, other molecules other than the virus itself. So there seems to be something which requires a coordinated attack, maybe of more than just natural killer cells. But
could be.
Okay, so an interesting question, which is more general but nevertheless relevant, is what are the main challenges that are faced by a more general application of cell therapy at a broader scale? What would you say? Is it scientific? Is it technological? Is it economical? Is it logistical?
I would say a bit of everything. Scientifically, we are progressing really extremely fast. In coming years, this technology will be, definitely available to a broader spectrum of patients. The other side is the economy, because at the moment the cost of a treatment is extremely high, and we cannot provide it to everyone simply because the insurance company couldn't afford it. It's extremely expensive. But by improving the technology, we are trying to lower the cost in a way that it could be provided almost to any patient.
And I presume that lowering the cost, what's necessary for that is to have these universal cells, right?
Indeed. For example, that. Yeah.
Once you have these universal cells, can you culture them to keep producing them almost as a?
Well, the cells they have certain capacity to divide. We can make them like immortal, but then we are risking the chance of having basically cancer derived from this immune cell, right? So let's say from one donor at the moment we can create a therapy for, let's say, 5-7 patients, which I think is still pretty good ratio. Right?
But the large expense of this therapy, and I presume this is costing governments around the world many tens of millions or hundreds of millions of dollars investment for its development. There's presumably companies also investing heavily into these treatments, potentially billions, I don't know. I can see potential future ethical problems. If you were to create a universal cell that could dramatically, almost overnight, reduce the cost to almost nothing, would that not mean a problem for the investors who've given so much, inputted so much into this future?
Well, there still will be some price, right? It won't be like nothing, because all the consumables are extremely expensive. But in principle, yes, there can be some economical issues, but I hope that that the right approach will win, and that there will be some. I mean, when you basically get bigger market, you can lower the price because it will return to you anyway at the end. Yeah.
But Michala, and this links to one of the questions about whether it's ready for use in patients who are suffering some form of cancer or not. Who should, in your opinion, who should be making the decision if we have a limited supply of such therapy? Costs are high. The complications of the right genetical modifications, whether or not universal cells are applicable or not, how far the hospital is away from certain centers like in Ostrava, etc., etc. Who do you think should make the decision, or who does make the decision about to whom goes this treatment and who not?
That's very difficult questions. I think it should be more directed rather to the clinicians than scientists. We are trying to provide a solution, and how is then basically transferred through the clinic to the patients.
Okay,
I mean, it's not on me. Yeah, I would like to give it to everyone. That this is my goal, right?
Yes.
At the end, but it's a very tough question.
Okay, so let's stick to that goal then. That it would be nice. I mean, it is obviously part of your vision that your successes and the success of others who are in the field with cell therapy would like to see this, of course, be available to whomever. So let's talk a bit about then. Let's end with a discussion of the future and how you see things panning out in the future. What can we expect in terms of cell therapy and its application in the future? So
it will be definitely more and more common. So nowadays it's applied only to maybe it's an answer to your previous question. It's provided to patients that they have no other choice. That all the current treatment were already tried, nothing works. So we basically give them the cell therapy because it's the last hope for them.
Right.
In a very close future, let's say in coming years, it will be given sooner to the patient, to bigger percentage of the patient, and in let's say 10 years and further, we hopefully will be able to target also the solid tumors, so different types of cancer, and the technology itself will dramatically improve in a way that we could modulate it at really quickly in the patient when the tumor will be changing, that we can basically the cells switch on, switch off, and like at the real time modify the specificity to hit the tumor and eliminate it really quickly.
Right at its beginning.
Exactly.
So that's almost like a sort of a vaccination type approach rather than it being a treatment.
Right.
Rather than when the patient comes already too late, a huge tumor, very difficult to attack. If it's a part of a sort of a vaccination approach broadly applied to the populace, then we could. Well, that sounds to me as we could potentially eliminate cancer.
Oh, this is probably too optimistic, but I don't know whether in our lifetime, but maybe our children, grandchildren could say something like this.
Well, fantastic.
Hope so. So,
well, Michal, any other aspect of your personal look into the future? I mean, so will there be another addition to the family? How about the farm?
Yeah, we hopefully will expand the family and also the farm and get some more animals, plant more trees, and yeah. Also, we are planning to renovate the institute, build maybe some new fancy building, attract more scientists, make the Ostrava great again, and modern and lively city with a lot of academic life and ultimately helping the
bubbling with the intelligentsia and activity. And well, Michal, I wish you all the best of luck, both on your farm, with your family, and of course in your professional career, and in Ostrava, and developing with your team cell therapies, which will be playing an incredibly important part in all our futures. And Michal, thank you very much indeed for coming on Global Science Café Czech Centers.
I thank you. It was a pleasure.
Yes.
Thank you.
Great to meet you. again, of course, future ones, which will be coming very soon. Great.